Hello everybody. Today we are going to discuss about phenyl alanine and tyrosine metabolism. Synopsis. This is the most essential heading that everyone should have in an essay for better marks. Here we discuss phenyl alanine metabolism steps, tyrosine metabolism, catabolism, metabolic disorders like phenylketonuria, clinical application of tyrosine derivatives. The subheadings we mentioned in synopsis can vary depending upon the type of question. For example, if it is like brief about conversion of phenylalanine to tyrosine and describe in detail about phenylketonuria, we need to mention the metabolic steps of phenylalanine conversion and also phenylketonuria. Only if we have more time, we need to write about the remaining aspects of tyrosine and the other metabolic errors. Whereas in the other question, when we are asked about metabolism of phenylalanine and tyrosine and inborn errors of metabolism, we have to write about each and everything. So, phenylalanine first. It is an essential aromatic amino acid and converts as it is the phenylalanine to tyrosine conversion is an irreversible step. It is glucogenic and ketogenic as the tyrosine gets catabolized into fumarate which goes to TCA cycle and acetoacetate which is a ketone body. Tyrosine is non-essential aromatic and has a sparing action to phenylalanine, which means that if tyrosine is part of our diet, then we don't need to be concerned about whether phenylalanine is there or not. Phenylalanine metabolic steps. Phenylalanine converts into tyrosine by phenylalanine hydroxylase, which later turns into para-hydroxy phenyl pyruvate by tyrosine transaminase, which again convert into homogenetic acid by dioxygenase enzyme. This HGA under homogenetic acid oxidase action changes to mallyl acetic acid, which changes later to fumarate acetoacetate by cis transisomerase. And finally, we get two products, acetoacetate and fumarate, by fumarate acetoacetate hydrolase. The key features of various steps include step 1 has NADPH, NADH and tetrahydrobiopterin as the coenzymes. The enzyme here is a monooxygenase, that is, it will incorporate one oxygen atom only into the product. So what we get is OH and H2O. OH goes to the product and H2O will be a byproduct. You will be wondering where hydrogen is from. It is from THPP and the THPP is replenished by the reducing agent of NADPH. Second step is transamination, where pyridoxal phosphate is the coenzyme and it is induced by glucocorticoid. Also, the third step is a dioxygenase catalyzed step and pyridoxal phosphate is the coenzyme here and vitamin C induces it. Now we check the important medical products from tyrosine. They are catecholamines, thyroid hormones, melanin and tyramine. Catecholamines. The epinephrine, norepinephrine and dopamine fall in this group where this epinephrine is a hormone while the other two are neurotransmitters. Now the synthesis of dopamine happens by dopaminergic neurons while adrenal medulla is supposed to synthesize epinephrine and norepinephrine. Functionally, adrenaline will be tachycardic and thus it is needed as an emergency drug in surgery. Then epinephrine causes smooth muscle relaxation in bronchi. Adrenaline is anti-insulin in nature and it is also the fight and flight hormone. Then the regulatory step in this metabolic pathway is one which is catalyzed by tyrosine hydroxylase. Here, the enzyme is allosterically inhibited by dopamine and norepinephrine, which means that they are non-competitive inhibitors. Then melanin. Melanin, as you all know, gives the black color to the skin, the hair, and it is seen in the eyes in the ciliary body, retina, and choroid plexus. Its function is to protect from UV radiation. While tyramine, as the image suggests, it is a tire with red color. This will denote that it is needed for increasing the BP. It is synthesized in the bacterial gut. Thyroid hormones, they are formed by the iodination of the tyrosyl residues into MIT and DIT, which later form T3 and T4. Melanin deficiency manifestations. There is a copper deficiency which results in fag type of hair, where there are alternate black and white hair. Then there's a condition of leukoderma, where white patches are seen because melanocytes are absent only in the skin. But if we take the case of albinism, melanocytes are absent throughout the body. Then graying of hair and malignant melanoma are other manifestations which you can learn. Then metabolic disorders. This flowchart we see here is a must. Phenylketonuria. It is caused by phenylalanine hydroxylase deficiency, and genetic mutation is if the enzyme is causing the enzyme to be not synthesized or to be non-functional. It is autosomal recessive. All the cases here are autosomal recessive. Frequency is 1 in 10,000 births. We see that it is of type 1, 2 and 4 and 5. Note that there is no type 3. Also, dihydrobiopterin can exist as tetrahydrobiopterin also, where tetra means 4. So it is in chromosome number 4. Also, if we spell out phenylalanine, we will get 13 letters. And so we can connect it with the idea that it is belonging to chromosome number 12. Alternate pathways. If suppose there is metabolic error and enzyme deficiency, phenylalanine will take other pathway to form pyruvate, lactate and acetate, which will accumulate and cause lots of clinical manifestation like low IQ, hyperactivity and agitation, tremors due to phenylalanine accumulation, then mousy odor and hyperpigmentation. The pictures are sufficient to remember. So the lab diagnosis. Blood phenylalanine is checked. Tandem mass spectroscopy is the most reliable test, but it's very costly. Guthrie test is a rapid test done on urine, and ferric chloride test is where the urine of patient is tested for phenyl ketones. It is a less reliable test. DNA probes are the most advanced ones to know whether there are defects in phenylalanine hydroxylase and dihydrobiopterin reductase. Now the treatment. We have to detect it early because every 10 week 5 units of IQ are getting lost. And the treatment is by providing a diet with low phenylalanine. Food supplements like tapioca can be of use here. The patients are advised to have a lifelong compliance to this special diet, but it's not mandatory. But in the case of a pregnant woman, to avoid the child from getting brain developmental defects, they need to have the special diet. Other disorders are like type 1 tyrosinemia, which is due to deficiency of fumarate acetoacetate hydroxylase and causing DNA damage. The symptoms will be like cabbage-like odor, hypoglycemia, urine containing tyrosine, etc. And treatment is by protein-restricted diet. Type 2 is oculocutaneous. As the name suggests, there will be painful corneal lesion, ketosis of palmar surface, etc. and low protein diet has to be followed. Type 3 is seen in neonates. There is transient hypertyrosinemia, and vitamin C help in improving the condition. So finally, to conclude, we need to write synopsis, diagrams, have flowcharts, and we need to be to the point. Need not be elaborate in the case of biochemistry. All the very best guys, do well for the exam. Thank you.